today I will be talking about AI in neuroimaging. And the basic purpose of today's presentation would be to give you an idea, the audience, the idea about what the neuroimaging is, what is AI. And I will briefly talk about two of my projects, but not the details, just in order to have an example of how AI can be used in neuroimaging for different kind of issues and to solve different kind of problems. So this is my brief introduction, but I will skip it because Salar has already explained everything. So, as I just mentioned, there are two things that I will be talking about, AI and neuroimaging. So first of all, let's start with what neuroimaging is. This is a very short, I think 1 or 2 minutes video.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss neuroimaging. I will cover a few of the most common types of neuroimaging: CAT scans, MRI, PET scans, and fMRI. Some neuroimaging techniques allow us to see the structure of the brain, while others allow us to look at brain activity or function. Computerized tomography, also known as computerized axial tomography, is a type of structural neuroimaging. It is usually called a CAT or CT scan. It involves taking a series of X-ray images from various locations around the head. These images can then be combined to construct an image of the brain. The resolution of CT images is not that high, but they can visualize any major structural problem of the brain, like a tumor. Magnetic resonance imaging, or MRI, involves applying a combination of magnetic fields and radiofrequency energy waves to the brain. Hydrogen atoms respond to the magnetic fields and radiofrequency pulses by emitting energy. The MRI machine receives this energy and can tell what part of the brain it came from. A computer can use that information to reconstruct an image of the brain that has high spatial resolution. Positron emission tomography, or PET scanning, is a way of imaging brain function. To do a PET scan, a patient is injected with a radioactive substance that emits positrons, which then emit gamma rays when they collide with electrons in brain tissue. These gamma rays are detected by the PET scanner. Because the radioactive substance was injected into the bloodstream, what the PET scanner is detecting is the movement of blood throughout the brain. Blood flow to an area of the brain increases when that area is active, so PET scanning creates an image that highlights the areas of the brain that are being used most while the person is in the scanner. Functional MRI, or fMRI, uses a similar approach to MRI, but focuses on the different responses oxygenated and unoxygenated blood make to magnetic fields and radiofrequency energy. fMRI uses what is called blood oxygen level dependent contrast, or BOLD, to identify changes in blood flow in the brain, and thus to identify areas of the brain that are most active. fMRI allows one to image brain function without having to inject anything, and provides high-resolution MRI images at the same time as it provides a functional image.
Okay, okay, as you just must have noted in this short video, there are a number of neuroimaging techniques that are used for either capturing the functional information or the structural information of the brain. The two techniques that I will talk today and I work with are MRI and functional MRI. So I will very briefly talk about the differences between these two, and then I will talk about how we are using them, or how you can use them in your studies. So the left side is showing MRI image. This is a 3D image of the whole brain. And in this image, what we can see, there are certain volumes, 3D volumes, small volumes that are shown in different colors like blue and green, and these are actually called the voxels of the brain MRI image. And these are, you can say, elementary units of the image. And in this case, in the case of MRI, the purpose of taking the scan is to have an image that has different colors, different resolutions, different intensities to show different parts of the brain, the structure of the brain, for example, gray matter, white matter, and the fluid that is inside the brain. On the left side, on the right side, on the other hand, we have fMRI images. Now, fMRI images actually capture the function of the brain. And as you can see, the main difference that you can see instantly from these two figures is that MRI is just one volume and fMRI are number of volumes span over time. So what actually happens in fMRI is that we use the same machine as MRI machine, but in a different manner. And we capture three-dimensional images over time. And that the time is actually the time of the experiment that we are working on. Like whatever we are doing, it spans the whole time. Again, the basic unit of functionality in a functional MRI is again a voxel, but there is a difference between the voxel of an MRI and fMRI in the sense that the resolution of the MRI voxel is a lot bigger, larger as compared to the functional MRI images. However, still the functional MRI is especially more dense, or it has a high resolution as compared to the other modalities that are used to capture the function of the brain, like EEG. And since the functional MRI images are 4D images, so in this case, we are not concerned with the structure in the data, but we want to know how the activity of different regions of the brain is changing over time, or we are concerned with co-activation of different regions in the brain under different conditions. For example, if I am talking what is happening in different regions, which regions of the brain are activated at the same time, if I'm looking at something, if I am listening to something, what is happening in different regions of the brain, that is what we study in functional MRI. And for that case, what we normally do is we take voxels and we extract the intensity level of those voxels over the whole time period, and that actually constitute the time series, and we deal with these time series in functional MRI. Now, this is about neuroimaging. Now, the next thing that I would like to talk very briefly about is what AI is. All of you must have some idea of AI, but I'm not sure what levels of AI do you know, but I will talk about the one that I will explain till whatever I would be using in this presentation. So actually AI is a science that is devoted to making machine think and think and act like human. So we actually want to mimic human in AI. And machine learning that we already mostly talk about is about making the machine learn how to act like the human. Without intervention of some programs, or without really explicit intervention from us. And machine learning is a subset of AI, and deep learning is another subset of even machine learning, which is again machine learning, but this is a machine learning part of the machine learning that uses a specific type of machine learning algorithms, which are called artificial neural networks. Now the question is, how do the machines actually learn? And for that, I will actually give a very, very basic example. Machine actually learn the same way that we learn. Let's suppose that these are all the pictures of cars, and we have bikes, and we want a kid to learn about the differences between a car and a bike. So what we would normally do is, if I am showing the pictures to a kid, I would, there can be two ways I would want the kid to, I would let the kid to understand. One way can be that I find some specific features of the car and the bike, which are different in both of them, like size of the wheel. And I tell the kid that, okay, if the size of the wheel is big, it is a car, it is a bike, and otherwise it is a car. But there can be someone else who can say that, no, this is not a good difference. So maybe the difference can be, instead of size, number of wheels can be a good distincting point. Someone else can say that, no, maybe color is a good point. The thing is that all of these things are features of these two specific classes of vehicles. But which one is better and which one is not good depends upon who is telling the kid what to look for, because the color is not, we know that color is not a good distinction because both of them can have the same color, but maybe the size of the wheel or the number of the wheels are a better choices. These actually distinct things are called features of these two distinct vehicles. And if we are telling the kid right features, then the kid would be able to understand the difference between these two things very clearly. However, another method can be that I just keep on showing the kid the pictures and just tell him or her that this is car, this is bike, this is car, this is bike, this is car, this is bike, and let the kid learn on their own what is happening. And that is called learning by not having any features. And so what do you think, which method should be the better one? Any ideas? No? Okay. So actually, so the thing is that there is no better or worse thing. If we are telling the kid specifically the features, then it means that we are letting them focus on certain things, and we would need less amount of data to learn about the distinction in the two categories. However, in that case, it also depends which features I am actually extracting from these two things. But on the other hand, if we are giving just a number of images and ask the kid to differentiate between these two things themselves, then in that case, they will extract the features themselves, and it may be easier for us because we don't have to have the features that are very distinct features, but it will need a lot of data. So the thing is that these are the two ways in which we make the machine learn, and these are the two ways in which we actually learn also. Then again, another thing that is important with reference to learning is what are the types of learning. Again, in our life, if we take the same example of car and bike, and we want the kid to learn, there can be two methods of doing that. One can be I label the car as a car. I tell the kid that this is a car and this is a bike. And whenever there is a picture of car and whenever there is a picture of bike, the kid would know that this is a car, this is a bike. And whenever a new picture will come, he or she will be able to classify explicitly which category the new picture belongs to. So this is actually the learning process, that we are training the kid on labeled data. And then based on the learning, they are able to classify new things. On the other hand, another method can be that we just show them the pictures and tell them that these are two groups. We don't name those two groups, we don't label those two groups, and we ask them that, okay, just divide them into two parts. They will do clustering, but in that case, whenever the new picture or new picture will come, they will not be able to know what that picture is. They will just say that, okay, it belongs to this cluster or this cluster. In most of the cases, again, both of them are right. There is nothing not right or wrong in this case, but it depends upon the type of the problem that we are talking about. Today what I will talk about is classification, and classification is called supervised learning. Clustering is called unsupervised learning. In supervised learning, in most of the cases, for example, let's suppose that we want to identify a tumor and non-tumor region. In that case, this is important that we have classification, or we have labels of different types of, like this is tumor, this is non-tumor. Just dividing it into two parts or three parts is not something enough. But in certain cases, it does not matter. So, but we can learn by classification or clustering, and there is another method which is in between, which is called semi-supervised learning. Okay, so now we have a bit of idea of what neuroimaging is, what AI is. Now we will talk about certain applications, and this is actually two projects that I have took from my lab, which are using neuroimaging and which are applying AI on that neuroimaging data to learn some aspect of the data. The first project that I will talk about, I will not go into the math of the project, I will not go into the details of the project, I will just talk about how AI is applied on neuroimaging data in these projects. So the first project actually was deep learning analysis of gliomas. I think maybe you would know, because all of you have, most of you have medical background, so maybe you would know better than me what the gliomas are. And now you know what the deep learning is also. This is a branch of machine learning that deals with the neural networks. So what we actually did in this project was that we analyze gliomas using deep learning. So this is just simple. And why did we do that? Okay, so we started this project in 2019. And at that time, one of my students who worked on this project, Maria, who is a PhD student, who is about to finish her PhD, she wanted to do something on brain tumor. And the reason she wanted to do something on the brain tumor was because she could find a lot of data available for brain tumor, and she felt that this is something that is important, not only for the whole community, but maybe from the perspective of Pakistan also. She wanted to do something on local data, but unfortunately we could not find the local data. Another thing that was issue was that because we didn't have the local data, we started to work on open access data. And with the open access data, it was difficult to find what question to answer, because when the data is open access data, a lot of people are already working on that data. So what she actually did was she started by reviewing all the studies, and she reviewed the studies from 2015 to 2020. She published a very comprehensive literature review on all of those studies. And in those studies, she explored what are the different AI algorithms that has been applied on brain tumor data, and what are the limitations of those studies. And at the end of this review, what we found was that there were a lot of studies working on different aspects of this data, for example, classification, survival prediction, segmentation, but there was no study which was taking into account all of these aspects together. So what we actually did was we thought of applying all of a method that can combine all of these aspects together in one framework to do all of the three tasks like classification, segmentation, and survival prediction on the data that is obtained from a brain tumor neuroimaging. And the end goal that we wanted to do was to develop a clinically accepted model for this whole data set. And in order to validate our idea, we actually talked to a number of radiologists in Pakistan. We talked to 10 radiologists, and we proposed, we actually presented our idea to them that we want to do something using AI on MRI data to have one framework that can classify, segment, and do the survival prediction on that particular data. And we got good response. 80% of them agreed that it is a good idea, and they thought that it would be good if we can develop something for them in the clinics. So this is a simple block diagram that we actually came up with. And the idea is to just take, this is actually the branch or the machine learning or AI that we are using here is the one in which we don't extract the feature from the data. So what we actually can do, or we did in this particular case, is that we took all the MRI images. These were input to one AI model, and then that model could do some processing separately on the three classifications, on the three specific targets like classification, segmentation, and survival prediction from the brain tumor. Okay, so in this case, the first thing that we had to have was the data itself. And we decided to use the data from brain, sorry, I cannot see that. Okay, sorry. Yeah, so we actually decided to use the data from a brain tumor segmentation challenge. And this is a challenge that is in progress for more than 10 years now. This is a challenge which is started in 2011. And the main purpose of this challenge was to segment different parts of the brain tumor. And from 2011 till 2022, they have been providing different data, which is collected from different institutions, mostly in US. And all of this data is pre-gliomas data. And the main purpose of this challenge was to segment different parts of brain tumor. There are 4 types of images that they have provided: T1, T1 weighted, T2, and FLAIR. And again, I would not go into the detail of these images, I would just like to tell you that these are all MRI images which are taken in different manners. And these are all providing the information about structure of fMRI from different angles. The next thing that in addition to these images, they also provided annotations by expert. So these are different parts of, there are 4 parts of brain tumor. That is important. I think Dr. Athar would know it much better than I do, that there are 4 parts of tumor like edema, non-enhancing part, enhancing tumor, and necrosis. And segmentation of these parts is important because these are important to identify in order to do surgery or any other treatment on the tumor, because they actually inform us about the level of or malignancy of the tumor or spread of the tumor in the brain. And the resolution of these images, MRI images, were 240 by 240 for every slice, and there were 155 slices for these images that were making one MRI volume. We used the data from BRADS 2019, which had 276 patients data. 66 of them were low-grade gliomas, or you can say low-grade tumor, and the rest of them had high-grade tumor. And the other data set that we used was BRADS 2020, and it also had 66 low-grade gliomas and 302 patients data available. After selecting this data, so this is the picture that is showing different parts of the tumor. As you can see from the left side, we have the whole tumor that contains all edema and non-enhancing and enhancing and necrosis, all the parts that I mentioned in the previous slide. And after the whole, inside the whole tumor, there is a tumor core, which is actually the part of interest for the surgeons. And inside that core tumor are the parts which are more dangerous or less dangerous. And these are the segmentation of different parts, as you can see from in the rightmost figure, that there are different colors which are showing different parts. And as I mentioned, all of this processing that is done in this case is classification, which is supervised learning. So what actually happened was that these data set has labels provided by expert about different parts of this tumor. And our goal was to use AI on these images in such a way that we could automatically extract these segmented regions. And for that, we did 2 steps. Actually, we did 2 different kind of processing. What was modeling on 2D data, and the other one was on 3D data. And how we did it was, and the first part, which was a bit easier, was that the input to the system was given in the form of 2D slices of the images instead of the whole images. And in this case, we use only high-grade gliomas or the slices from the tumors which were a grade 3 or grade 4 tumor. And the end goal was to classify those slices in tumorous slices or non-tumor slices. Again, as I told you, that the main purpose at the end for the whole study is to come up with something that is very easy for the clinician to use, and they can do basic tasks that they do manually right now, not only 1 task, but 3 tasks in one go. So in this case, the first purpose of this project or this study was to come up with something that can differentiate the slices as tumorous or non-tumorous slices. And the other thing was the same, a segmentation of different areas in the tumor itself. This is the network that we use. Again, I will not talk about how this network or details of this network. I would just like to tell you how can this AI model be used to identify or to segment and classify in the same model. So what we did was we gave the input to the model in the form of 2D slices. And you can think of actually these 2D slices as images of cars and maybe bike. And so we can actually imagine that the non-tumor slices are cars and the tumor slices are bikes. And at the end of the day, our goal can be the model that can classify cars and bike successfully. And in addition to that, it can segment part of those images also. For example, we want to extract just wheels from the car or wheels from the bike. So in that case, what is happening is that the first part that is enclosed in this black box, black square, is actually the part that is called encoding part. And what this part is doing is that it is taking the images and it is passing all of the images through number of parts. And at the end of this, it is extracting the information from those images about different parts of the images, and including the part that is the target that we would like to segment. For example, the wheel of the images, or in case of a brain tumor, the tumorous part that we would like to segment. And it encodes those parts and keep the information with itself. And at the end, what it does, it gives us the classification of the two types of images, normal or abnormal, or like car or bike, if we talk about the other example. And so this is one part that this is doing. The other thing that we want it to do is segmentation. And the next, you can see that actually what is happening in the beginning, we have these 3 blocks, which are encoding the data. And the first 3 blocks make encoder, that is giving us the classification as the output. The last 3 blocks are decoder, and they are actually doing the reverse of whatever encoder has done. And in the reverse process, they are again generating the same image. However, this image has that segmentation information enhanced. And how could both of these encoder and decoder combined do that? Because in the input, we did not only provide the images themselves, but we provide the information about segments also, about where that brain tumor is and different part of the brain tumors based upon the annotations or labeling that we obtain from experts. And based on that, in the case of encoding part, our algorithm could learn how to differentiate those parts from the rest of the image. And at the end of the decoder part, it could actually give us the segmented output. Okay, so this was the end, and this is actually just one representative slides of the result. In the very left side, we have original image. Center is the ground truth. Ground truth is actually the extracted segmentation of the brain tumor based upon whatever the expert radiologist provided. And the last panel is giving us the information about, the last panel is actually the result of our own model segmentation. And usually you can see that, okay, they are not identical, but they are very similar. We can see the same colors in both of them, and we can see the same sizes also. But we did not do it only on one image, but we did it only on all the images that we were working on. And then the next thing was how to decide if this was a good enough segmentation. And one of the ways to do that is to calculate DICE score. Again, I will not talk about what DICE score is, but I would just like to actually emphasize here that we use AI models a lot in these kind of problems, but we cannot use them blindly. There are certain parameters that we need to not explore actually, we need to calculate the efficiency of the different things also. And the efficiency of classification is measured in terms of accuracy, which is just telling how accurately the 2 or 3 or 4 classes are identified as correctly by the model. And the efficiency of segmentation is measured using DICE score, which tells us how good the match of the labels provided by the radiologist or by the expert is with whatever we have obtained from our own model. And in this case, you can see, and again, another thing that I would like to mention here is that AI is a lot hit and trial method. If you ask someone that what is a good model for any specific problem, that person would not be able to tell you directly. The reason is because in case of AI, we tune a lot of parameters and we keep on training and testing and keep on obtaining these scores, accuracy and DICE scores, and then other measures for other kind of problems. And we report the things that are giving us the maximum or the best performance. So in this case, you can see I have highlighted something in red, which is for BRADS 2019. We have 99.71 accuracy, 93.63 score for DICE score. And then we have DICE scores for different parts of the brain tumor, like whole tumor, tumor core, and then enhancing tumor. And all of them are giving the best thing that we are obtaining. We are actually reporting that. And same thing is true for BRADS 2020 also. So this was, so what we actually found from here is that, okay, the model that we selected was doing pretty well on 2D multitask learning. The next thing that we did was we moved to 3D multitask learning, which means that the input now, instead of slices, we were giving the whole volumes as the input to the model. The model is exactly the same. There is nothing different. There is a shared model here. And then after the shared model, there are task-specific layers. And in this case, what we were doing was, again, classification, segmentation, and survival prediction. But in this case, for the classification, we were not classifying the tumor and non-tumor slices, but we were classifying on the basis of volume, whether they were high-grade tumor volumes or low-grade. And by volume, I mean the whole MRI 3D image. And segmentation was again the same, but in the volume, and survival prediction was how many days a person would be able to survive, how many days do we predict the person would survive based upon our model. Okay. Now, again, I will not go into the detail of what is happening. I would just like to emphasize that switching from 2D to 3D was not that difficult. It is exactly the same idea. The only thing that is changed is how are we building the input block and what are we expecting at the output. So in this case, because it was 3D, so what we did was we actually input 3D volumes, and then we wanted to have classification between high grade and low grade. So with each volume, we attached a tag, which was again given to us by radiologists, whether this tumor was high grade, low grade, low grade. And then in addition to that, we provided the segmentation, again, the same thing that we provided previously. And in addition to that, with every volume, we provided the information about what was the number of days that the patient survived after the tumor diagnosis. And that information should help our model learn to predict the survival of anyone who has tumor, a patient who comes in the system, a new patient, and the system should be able to correctly or approximately correctly identify how many days would the person survive. So again, this first part that I am showing enclosed in the red block, this is the encoding part. It is again exactly doing the same thing, but based upon the data, new data that we have provided here. And from this part, we are getting two things from the encoder, as you saw in the last case, that from the encoder, we were getting classification. Here again, we are getting classification and survival prediction, both from the encoder. And from the decoder part, again, we are getting segmentation. So what I actually want to emphasize, or what the reason that I discussed both of these scenarios, is just to tell you is that AI is not that difficult to work on. If you have the right amount, right kind of data, automation, doing automation is not that difficult. And the things actually, all of these layers and everything that are in between, they are mostly hit and trial method, and whatever is giving us the best performance, we actually store that information, and then we work on that in future. Now, this is again one slide. I have talked about this previously, so I'll not go into it. And these are the results. Again, there are a lot of results. I will not actually go again in the detail of those results, but what I would like to emphasize on is these red blocks. And you can see we didn't have good result in this case. We have actually, this paper is again under review. The previous paper has been published. This is again under review, but we did not get good results. For example, here you can see that we classification accuracy, we are getting 75% only for BRAS 2019. And then here, if you look in the other side, the DICE scores are also not that good. And the reason being, though we are using an identical algorithm, not identical, but the same principle as we did before, but we are not getting very good results here. And the main reason is that in case of 2D segmentation, we were giving slices. So every 2D segmentation and classification, we were giving slices. And there were many slices. In one volume, there were 150 slices, 55 slices, and then 155, and then 155. So for 2D classification or 2D problem, our model had a lot of data available. But in this case, when we are doing the things based on the volume, we have only either 200 something or 300 something data available on the two data sets, which is not enough to classify or to do any of these things very reliably. And then another reason is that in the case when we are using so much data and we are combining all of these modalities, the error is mostly also accumulated. The error of survival prediction and classification and segmentation is propagated together, and it is not easy. And that is maybe one of the reasons that the people have not actually done this multitask learning till now for these problems, because it is not easy to start adding up different tasks in the same model, though for a few tasks it can work very well. And again, this is the result for survival prediction. I will not go into the detail, but what I would like to conclude from here is this is one way AI can be used on MRI images or neuroimaging data. And I would like to emphasize on the fact that the same things may not work so well on all kind of data sets, brain that are supposed to be activated during emotional processing. And we combined those time series with the labels that we generated during sentiment analysis, and we provided this information to different classifiers. We use machine learning classifiers, we use deep learning classifiers, and we classified the fMRI data using the labels from another different genre subtitles. And we got very good results. The combination of one of the sentiment analyzer and one of the classifiers gave us accuracy above 90%. So I will just stop. I would just like to conclude from here. I will just have two more slides, which are about what were the limitations for the studies. Again, I just wanted to give you a flavor of how many ways the AI can be used in neuroimaging. And we got good result in certain cases. We did not get good results in certain cases. And the major limitations in most of the cases that we came across as computational resources. For example, in the case of brain tumor data, we had to reduce the size of the images in order to process because we did not have computational resources. Similarly, in the other case also, we could use only 30 minutes of movie data because we did not have computational resources. Then again, the funding is an issue due to which it's difficult to keep the students on the same project even after completion of their degree. Another thing that is actually not very encouraging is that we don't have local collaborator in terms of acquiring the data. So we have to rely on open access data set, and it has its own limitation. Like I just told you that there are no labels available for whatever we want to do. So this is something that is that limits our extent of what we want to do with AI in neuroimaging. These are some future directions that we would like to continue on for this project. As I told you, we would like to improve the result of our first study, the brain tumor study, for the 3D analysis part. And at the end of the day, we would like to have an app that can assist the clinicians in real time. And for the other parts, it's for the other study, it's just a start. We would like to extract the label from speech, music, images, and so forth. And we would like to apply those images, those labels on the data and see how it works. These are my grad students. I have 4 female grad students and 2 male grad students. Maria and Rimsha actually worked on the project that I just talked about. And all of the rest of them are also working on similar kind of project with naturalistic stimuli or MRI data. This is my email address and my web page address. Thank you.